All right, so we're going to look at cell division. Biology is the only time that when you use the term multiplication and division, it actually means the same thing. Our cells multiply, meaning getting bigger, by dividing. So the individual cells divide to multiply our numbers. We're going to look at the cell cycle, cell growth, cell division. When you first started, you started as a human egg and a sperm, and when they fuse together, they are the size of the egg, which is roughly the same size as the period at the end of a sentence, okay, as a period at the end of a sentence in your normal average textbook. That means a 12-point font, period. That's the size of a human egg. Now, after that, we have to have all kinds of cell division happening. To go from an egg to a baby, we have fertilization, and then divide and divide and divide and divide a lot of times. So cells divide for several reasons. Reproduction, and we see this with asexual reproduction in our single-celled organisms. They divide for growth, okay? To go from a fertilized egg to a multi-celled organism, we need lots and lots of growth. And for repair and renewal, replace cells that die or normally wear out. And basic like injury, if you get a cut, we have to fill in that space, means we need to make some more cells. So here we see an amoeba, it is dividing in half. This is just kind of a quick image of cell division. You start as a single cell and we divide and divide and divide. Here's some growth that kind of went a little wonky. And as you know, things like lizards can grow their tails back. So here we see it in the midst of growth. And I don't know what happened to that picture. Oops. Now we have our original cell, and when it divides into two, we call those each daughter cells. Okay. So what is passed to these daughter cells? An exact copy of the genetic material, or DNA. We want every cell to have the exact same DNA as the original cell. That way it has all the instructions, can do all the jobs it needs to do. Now you can see here at this image down below, we have, this is an epithelial cell, which means skin cell. And it's taken from a kangaroo rat. And we can see the chromosomes inside, the orange, and all these green little fibers, those are our cytoskeleton. So remember back to our cell structures. Now, this is when we get exact copies, we call it mitosis. And then we have to divide the organelles, cytoplasm, cell membrane, enzymes, all that other stuff that's not in the nucleus. We have to divide that as well. But that happens during what we call cytokinesis. Now you guys are going to have to remember back to 7th grade and 9th grade when you had cell division there. We're going to kind of review on that, but if you need more review, please see me during office hours so I can make sure that you're on the right footing for this, okay? This is what a kangaroo rat looks like. Notice its feet here, how big those are. Its leg goes way up here. It's designed so it can jump, kind of built like a kangaroo. So an overview of mitosis. You did this again, 7th and 9th grade. We have multiple stages for mitosis. We start with a normal cell, a cell just hanging out. And that we call interphase. This is not part of cell division. This is just the cell hanging out. Now, cell division starts with prophase, and in prophase, we pack our DNA. Okay, we pack it into chromosomes, and it gets thick. And if this was like, you know, this big bowl of spaghetti, now we're going to clump it into its individuals, and it has its replicated copy with it. We call these sister chromatids. Okay. This is called prometaphase, and we're not really going to worry about that. Nobody's going to talk to you about that. However, you will see it in images. It is in between prophase and metaphase. Okay, it's kind of in between. You'll see that the cell, the nuclear membrane is dissolved. We have our spindle fibers that have attached here to our chromosomes, and this in metaphase, they're in the middle. Now they don't line up perfectly. We'll see some, ends trickling, moving back and forth, They're not exactly in a line, but they're along the metaphase plate or the middle plate. During anaphase, they're going to pull apart. So we're going to separate our pair. So these are our identical copies. We're going to separate those copies so that one copy goes to each end. And then during telophase, we are going to be separating them, reforming our nuclear membrane, and cytokinesis is actually dividing them apart. So dividing them into two. IPMAT, this is what you need to remember, IPMAT, IPMAT. There's lots of different mnemonics. If you come up with a good one, let me know, I'll share it with the class, okay? So interphase, prophase, metaphase, anaphase, and telophase. Interphase is in between, prophase is packing, metaphase is middle, anaphase is apart, and telophase is two. We then end with cytokinesis where it cuts, okay? So here's interphase. And if we look at this image here, imagine the cell cycle like a clock. And we're going to spend a whole bunch of time in interphase and a little bit of time in mitosis and cytokinesis. It is in fact about 90% of the cell life cycle for most cells. And this is where it's doing its general everyday job. We reproduce the RNA, synthesize proteins and enzymes, and prepare for duplication triggered, if triggered. Okay. Once signal, then we go into the mitotic phase, okay? So we're actually going to do mitosis. Cells grow and mature and divide again, okay? That means that they're going to go through these parts of mitosis. So we have gap 1, S, gap 2, mitosis. This is actually when we have gap 1, S, and gap 2. This happens during interphase and then mitosis. Okay. We see this with epithelial cells, blood cells, some stem cells. So if we were looking at that clock again, all of this blue, green, and yellow, this blue, green, and yellow is all interphase. We break interphase down into little sections. Then mitosis. Now, some cells grow and mature, but never divide again. Okay, if that's the case, then instead of continuing through interphase all the way to mitosis, they stay in this first stage, gap 1, and we say that they go into a resting phase or G0, G0. Okay, this is basically, it's just going to hang out here, keep doing its job over and over, but it's not going to divide. These include very important cells in your body like mature muscle cells and mature neurons, mature nerves, mature brain cells. This is why if you have damage to your brain or your spinal cord, we cannot fix that because those cells will not regenerate. They can't be made to grow again. Now, depending on the type of damage, so for instance, if you have minor damage to the brain, sometimes the rest of the brain around it can pick up the slack and still help you out. Same kind of thing happens with muscle, but if there's too much muscle damage, then that can't be fixed. Okay, now we have some cells that are super special, and this would be our liver. In our liver cells, they actually can be resting, meaning not dividing, and then go back to dividing again. So they don't divide continually like our skin cells do, but they do divide when we need them. Okay, so here again, we have our cell cycle. You'll see about 90% of it is interphase. We break interphase into 3 different steps. We have cell growth. Okay, we call this G1 or gap 1. We have S, which is synthesis. This is where we synthesize the DNA. And then we have G2. Okay, this is our gap 2 where we have some more cell growth. Then we have mitosis and cytokinesis. So here for interphase, we have G1, which is gap 1 or first gap. This is where growth happens. For an average cell, it takes 10 to 24 hours in this stage. It's doing its everyday job. Now, during this time, it makes more proteins and organelles. So it started off with some when it divided, but now it needs more. as it gets bigger and grows to capacity, it's going to need more proteins and organelles. We also catabolize carbs and lipids, and our cell will double in size. Next, we hit the synthesis phase. This takes about 5 to 6 hours. So it takes about 5 to 6 hours to make a complete copy of all of your DNA. Our chromosome number is doubled. So when we have our original set, plus we make a copy, that means we have 2 sets. Our chromosome number has doubled, and we are what we call 4N. Now the N refers to the number of sets of DNA that you get. So you got 1 set from your mom and 1 set from your dad. So you have 2N. When we replicate your DNA, we take the 1 from mom and 1 from dad, and we replicate those. Now I have 4, so you are 4N. Now this is gonna be more important in our next unit when we start talking about genetics, but just kind of a preview of what that means. Chromatin forms. Okay, we'll talk about that more. Okay, gap 2, G2. This is where we are preparing for cell division. When we prepare for cell division, okay, we are going to have more growth. Okay, we got to be able to have enough for everything to be able to grow and be able to have enough that the new cells have enough to survive. Okay. We produce more proteins and more mitochondria. Okay, why would it be important to make more mitochondria? Yeah, it gives us more energy. So it also means that when I divide the cell, that both sides, so both new cells, when I pull them apart, are going to get enough mitochondria to make enough ATP for the cell to survive. Again, lipids and carbs are catabolized. This word's come up twice. Catabolized, what does that mean? Remember, it has cat in it. Excellent. Oh, I love seeing that pop up in my chat there. Yes, catabolize means to break down. Cats like to destroy things. Okay, now, if it is a cell that is going to go into the resting phase, we say that's G0. It happens after G1. So from G1, instead of going into synthesis, it is what we call G0. And it just continues to do its normal activities, and it never actually goes into the S phase. All right, so we're going to talk more about what happens during the mitotic phase, but I want to talk about cytokinesis real quick, then you'll have the whole picture together. So cytokinesis is when we divide cells. Okay, so we're actually doing the we're talking about the cytoplasm or cell. We're going to divide the cytoplasm. So mitosis is dividing the nucleus, whereas cytokinesis is dividing the cytoplasm and everything inside the cytoplasm. And if we look at animal cells, it starts by having this part where it just starts to like squeeze in, and it squeezes in. We call that a cleavage furrow. Okay, if you look at the image down here, it is a cleavage furrow. Maybe you'll understand now why in common language we've used the term cleavage for certain things. Okay, so a cleavage furrow begins to form. A cleaver is a knife that has, remember those big square knives you'd see in like horror movies? that's called a meat cleaver, and it's designed to cut in half. Okay, so you can cut things in half. To cleave means to split. So we are splitting that cell into 2 new daughter cells. In plants, it has to happen a little bit differently because there's a cell wall. So in plants, the cell plate forms. And you can kind of see it here in this image, but definitely down here in our drawings. Vesicles line up at the equator. Now, our Golgi, so we have the Golgi in here, has been producing the materials that go here, okay, to make our cell wall. And remember that that is a bunch of things like carbohydrates in the form of cellulose. So we are making vesicles or bubbles that the Golgi sends, and they line up right in the middle. Now, if you've ever had to do dishes and you've ever sat and played with the bubbles, you know you can kind of get 2 bubbles and pop them together. Okay, a couple of you are looking at me like I'm weird. You know you've played with bubbles. I'm not the only one. Okay, so you can pop these bubbles together and they make one bigger bubble. Well, that's what's happening here. I'm popping bubbles together to make bigger bubbles, and eventually it makes a line and it goes all the way across. And then the membrane forms around so that I get 2 distinctly separate cells. Okay, so again, derived from the Golgi, they fuse together to form 2 cell membranes and our cell wall. Okay, so our cell plate forms, the vesicles line up at the equator, vesicles fuse to form our 2 cell membranes and the new cell wall in between. It's really kind of cool, but if you take a look at this plant cell as it's dividing, what do you notice about the size of the new cells? Good. Yeah, our new cells are significantly smaller than the old cell. This means that there is a point where they can't keep dividing, because otherwise I'd have little teeny tiny cells and they just aren't able to do the same job. So in plants, there is a limit to how much they can divide. Now, our frequency of cell division, how often this happens. It varies depending on the cell type. I mean, as we said, nerve cells, those don't divide. So the frequency is going to be like nothing. An embryo has a cell cycle of about 20 minutes. So once the egg and sperm fuse, that embryo divides about every 20 minutes. Okay. This means that, I mean, in 20 minutes, I go from 1 to 2. In 40 minutes, I go from 2 to 4. And in an hour, I go from 4 to 8. So in an hour, I go from 1 to 8 cells. That's actually really quite rapid. And then you can see how it's going to exponentially grow, because now that I have 8, my next division is 16, then 32, 64, and it's going to start going really quickly, very rapidly. It's actually within the first couple of hours after fertilization that if twinning is going to occur, that it occurs. Now, twinning is when at the very early stages, so usually within the first like couple hours, 2 hours, the cells will, as they're dividing, for some reason, instead of dividing and staying together, they divide and separate. And now these grow on their own to create twins. Okay, that's for identical twins. Fraternal twins are different. Our skin cells are constantly being renewed. They divide frequently throughout our lifetime, and they have a cycle of about 12 to 24 hours. About every day, you are getting new skin cells. Now, that's not all your skin, and your skin has multiple layers to it, but the very top layer is getting rubbed on and it's dry and it comes off. And so it has to be replaced. Now, weird fact, about 90% or over 90% of household dust is actually dead skin cells that have come off of the people that live there. So when you are dragging your finger across your TV and you get that like, yeah, that's like dead skin cells, all that. when you're having to, you know, mom makes you go dust the living room, that's mostly dead skin cells. Okay, kind of interesting. Question was asked, since cells become smaller when they divide, does that mean that there are different cell sizes in the body? There are, however, with animal cells, remember that our first step in our interphase was growth. So it will divide and be smaller for an animal cell, but then during that first G1, that first phase, they grow back up to what would be normal size. So in animals, they are small when they first divide, and then they go back to a normal size. They'll get slightly bigger before they divide so that the two new cells aren't too tiny, and then those grow back up. So in animals, we don't have that getting smaller and smaller and smaller because we don't have the cell wall to interfere. So yes, so like, let's say we're talking about your skin cells. So the bottom layer of your skin cells is where we're producing new skin cells. They will grow and then divide. And then like this cell will grow and then divide. And then this cell will grow and then divide. So it's this continual process of getting to the right size and shape and then divide, and then get back to the right size and shape and then divide. So it's a continual process. Now that's only in those particular types of cells. Okay. Again, we have cells that don't ever do that. So it just kind of depends on which cell we're looking at. Good questions, you guys. Now our liver cells, we said that those go into our G0 or our resting phase, but they still retain the ability to divide. And they do this to kind of help our liver. They divide about every year or two. Now, your liver is the part of the body that filters toxins out. So the liver is specifically designed to pull out poisons and toxins from your blood. Okay, that's why if you drink alcohol, it can damage the livers because your liver is having to pull all of that out. Medications can cause damage to the liver because your liver is trying to pull what it sees as a toxin out of the bloodstream. The liver it's pretty much the trash dump of the whole body. All right. And that's for all animals that have a liver. The liver is a collection of all the toxins and nastiness. So because it's collecting all that, some of those cells do die, and we do want them to be replaced. They get worn out. So about every year or two, we replace those. Mature nerve and muscle cells do not divide after they reach maturity. They are never going to divide again. That's why it is so important to be careful with what happens, especially to your brain and spinal cord, because once that's damaged, you don't ever get that back. If we could figure out how to stimulate the regrowth of that, not to mention Nobel Prize, but we would be able to help people who have like spinal injuries be able to regenerate that, be able to walk again, have any kind of damage, accidental brain damage from things like automobile accidents, falls, any of those things, we'd be able to regenerate. That would be pretty amazing. They're actually using, looking at the mechanisms in things like starfish and lizards, the things that do regenerate, to see how we might be able to kind of use that for us. We're not there yet though. Are conditions such as scoliosis partly due to cell division then? You know, specifically scoliosis, I'm not sure. It would make sense that it has to do with that. Usually there are other conditions involved and not just cell division. Usually we are looking at things like, the scoliosis deals with the bone and bone development. And so it could be just a depositing, where it's depositing of the minerals and it not being equal on both sides, which causes a bending. Okay, but I would have to look into that. I will try to do that. Let me make a note real quick. Scoliosis. All right, I'll look into that. Sorry, I don't have a better answer for you. All right, checkpoints. We have throughout the system checkpoints, and these are just like stops. And it's like a stoplight. We check in, say, hey, is everything good? All right, go ahead and go. Okay, if any of you have ever been go-kart racing, if you've ever been to like a roller coaster, you get into a roller coaster, you buckle in, and then they come and check, and then they're like, okay, you can go. We have checkpoints like that. And if things aren't what they're supposed to be, then we can't go. All right, so we're gonna kind of look at how that's regulated. Signals indicate if the key cellular processes have been completed correctly. And you'll notice here, these little gates, we have 3 different checkpoints or 3 gates. And our first one, so if we always start with G1, our first one is going to be here at the G1 checkpoint. This G1 checkpoint checks for nutrients, growth factors, and DNA damage. So here at checkpoint 1, so checkpoint G1, we're going to see, does the cell have all the nutrients it needs to be able to divide? If it doesn't have enough nutrients, then when it divides, the 2 new cells will both die. That's a waste. Why do division if they're just going to die? That's a lot of energy wasted. Okay. We also check for growth factors. Have I gotten a signal that says I should divide? If I don't have a signal to tell me to divide, then I shouldn't divide. Okay. And then we look for DNA damage. Does the DNA have everything it needs in the right place? Is it solid? Is it made right? Or is there a problem? If there's a problem, we don't want this cell to replicate, because that means that both new cells are damaged, and that can cause problems for our body. Okay, now if I get to this checkpoint, everything's good. Check, check, check. Yep, you're good. And then I either go to resting state and I continue doing my job, or I can move on to synthesis. So these are almost like quality control. Yes, that's exactly what these are like. So these are just checking to make sure everything's right, everything's good. Okay, keep going. So good, that's a good analogy. Now we continue on to the synthesis phase. So I got to here, everything's good. I've got the signals, I've got enough nutrients to be able to do everything. And my DNA is intact and working properly. So I'm going to go ahead and move on to synthesis, where I get to make another copy of the DNA. And then I'm going to go into G2, where I get to increase in my cell size so I can get ready to divide. Now I'm going to divide after this. It takes a lot of energy to move all the chromosomes around. It takes a lot of energy to cut that cell in half. So I don't want to spend all that energy unless it's going to work for the benefit. Okay, so everything's going to work out right. So in this case, I'm going to check for cell size. Did it get big enough? Because if I divide and it's not big enough, both cells will die. Okay, and then I also check to see, did the DNA replicate properly? Okay, if the DNA replicated properly, both cells are going to be good. If it didn't, then one or both cell is going to have problems, not be able to function properly, potentially die. So I don't want to divide unless I have all that in there. And once I go, yes, all there, we're good, then I can go into mitosis. Okay, so I start mitosis, start with prophase, where I pack everything up tight. So I have my replicated DNA. I'm going to pack it all up tight. Okay, and then after prophase is metaphase. Okay, metaphase is where they all line up and I have the fibers attached to it so it can pull it apart. So it's going to check to see, are those there? That's my last checkpoint. If I don't have those there, then they won't pull apart properly and I'm going to have the wrong amount of DNA. So it's going to check, yes, I can pull apart, I'm good. It'll keep going. If it's like, no, those spindle fibers aren't there, they're not doing what they're supposed to do, it'll actually just recycle that cell. Be like, I'm done. Get rid of it, recycle it. So it's like checking before we sell it. Exactly. Exactly. So we're checking to make sure everything's right before we hand it off. Okay. What happens if it's stopped at the checkpoint and is not allowed to keep moving? Does it just stay there and die out until it's recycled? Yes. So if it hits a checkpoint and it cannot proceed past that, usually what happens is apoptosis. Okay, so if we hit that checkpoint, I can't go any further, it will usually trigger signals that will cause the cell to basically kill itself. And those parts, we try not to get rid of anything we don't have to in the body, so we recycle those. They're broken down, usually in the liver and along the way, it gets all broken down, and then we use those parts again. There we go. So in the G0 phase, again, not dividing, it is in its differentiated state. So this is a cell that has become whatever it's going to be, a heart cell, an eye cell, whatever. And it's just going to stay in that position and just keep doing that job. Most of our cells are in the G0 phase. Okay, most of our cells are just like hanging out, doing their job. Our liver cells can go here, and then they get called back by signals. Okay. So these signals are, again, damage to the cells. The cells actually, when they apoptosize, when they disintegrate, kill themselves, they will send signals out to other cells that can tell them, okay, you need to replicate because I just died. So we need to fill this in, okay? To clarify, the cell material is recycled after apoptosis. That is correct, yes. Now, nerve and muscle cells are highly specialized and they are in this G0 phase. We say it is arrested in G0 and can never divide. Now, the go-ahead signals. So we said that there are those 3 stops, okay? And these signals are ones that promote cell growth and division. We have external signals, we have internal signals. Our external signals are going to be growth factors, okay? Some type of growth factor. And this could be general body growth. And that means that it's a hormone that's sent through our blood, and the cells receive that from one of our receptors on the cell membrane and send a signal in that says, okay, it's time to divide. It could also be an internal signal, something in the cell that's been triggered that says, okay, time to divide. And we are going to look at these little guys. These are called cyclin-dependent kinase, okay? It's an enzyme, and we control turning this on and off based on having phosphorus attached to it, okay? Phosphorylation. You'll find that we've done phosphorylation several times already. ATP, when we add that phosphate to ADP to make our ATP, that's phosphorylation. Phosphorylation just means adding a phosphate. So we are going to phosphorylate this to be able to make these signals happen. We have what is called a cyclin. It's a protein, purple molecule here. So we have this cyclin, and we have our cyclin-dependent kinase. So this is an enzyme. And then this is another protein that's going to work with it. Okay, remember we've talked about how sometimes they have to work together with other things to turn them on or turn them off. Well, our cyclin is our regulatory protein this time. And the levels will actually cycle within the cell. They go up, they go down. How much cyclin is there, how much is produced, how much is broken down, it will fluctuate. And then we have our CDKs, cyclin-dependent kinase. So this is a kinase, an enzyme that depends on cyclin being present. So we have to have cyclin for it to do its job. These phosphorylate cellular proteins. It activates or inactivates the protein. Okay, so when this is here, this can tell the phosphates to be added or removed to other proteins within the cell. When we put these together, okay, we call this the CDK cyclin complex. Just like an apartment complex is a bunch of apartment buildings together, we just call it a complex, they're all together. So here we have a complex of our cyclin-dependent kinase plus the cyclin together. That's our CDK cyclin complex. It's also called an MPF. We don't care. You might see that in some of the drawings or any readings that you do. You are very likely not to see that on the AP exam though, okay? Look for cyclin-dependent kinase, CDK, and cyclin. Now these trigger passage through different stages of the cell cycle. Okay, these help regulate it. These are the ones that are like, okay, everything's good. Now we can hook up and let everything go. Now I said that the cyclins will cycle during the cell's lifetime. And we had scientists, oh, Sir Nurse disappeared. Sorry, my image disappeared. So these scientists all worked on this at different points in times. And you see that one scientist was able to find and kind of discover and understand that there are checkpoints. And then we have another scientist that found the CDKs and another scientist, different scientists that found the cyclins. So it takes a lot of people working together to discover these things. And this is why it is so important for us to publish and actually give our findings out to be able to share that information in science. So you can see here, it took 3 different scientists to be able to understand this. Now, they studied the cell cycle and would look at what stages the cell was in and then test to see when the cyclins were present at what levels and when the MPF activity was happening. So that CDK cyclin dependent complex, okay, when that was actually doing a job. They were able to see that it increased, the cyclin increased to a certain point, and then the MPF activity would start to happen and mitosis would occur. And when mitosis was done, they dropped. And then the cyclin would build back up, we'd start having activity, and then it would drop. And they found that this cycle was very consistent. Okay, so this was how we kind of decided or determined that these are cycling within the cell in conjunction with those checkpoints. Now, external signals. Again, growth factors. This is coordination between cells. Protein signals are released by the body cells that stimulate other cells to divide. And we have something called density-dependent inhibition. So we can get growth factors to these cells. And because they're not surrounded on all sides of the cells, they'll go ahead and divide. But once we hit the point where they're surrounded on all sides, they stop dividing, even if that growth factor is present. It'll stop dividing because it's surrounded on all sides. It's like going, you know what, there's not enough room, there's not enough space, we can't keep dividing. But if I remove some of those cells, it'll then again go back to dividing. Okay, so it kind of fills in the gaps. And you can see how here, if your skin were cut, then we need to fill that in. We also have what is called anchorage dependent. Anchorage dependence means it is anchored somewhere. Okay, it's able to have its anchor there and it's not like free floating. And anchorage dependence means that they have to be attached to something to divide. So these ones are anchorage dependent. They're attached to the bottom of the culture plate and they're dividing in here, okay, attached to the bottom. A lot of our cells have anchorage dependence. They have to be attached. Now here's back to our images. The images always make things a little easier for me to understand. I'm hoping they do for you as well. So you can see our cell membrane here on the outside, and here's one of our surface receptors. Now this is just a basic surface receptor. This is not a G-coupled protein. This is not tyrosine kinase. Remember, G-coupled proteins has that little slingshot dude, and then our tyrosine kinase has 2 that work together. So this is just a cell surface receptor. The cell surface receptor receives a receptor, and then we have transduction of the signal. And then within the nucleus, okay, we are trigger cell division. So we receive the growth factor. So in this case, growth factor can also be called what? What do we call those molecules that are received by the receptor? Ah, look at all of you on top of this. Oh, makes me so happy. Yes, we call that the ligand. The ligand. All right, so growth factors are just a signal. And in this case, that signal is received by that cell surface receptor. Okay, so ligand is that general name, like saying dogs. Growth factor is a very specific name, like saying, I don't know, St. Bernard. Good job, guys. There are some examples of growth factors. We have platelet-derived growth factor. Okay, platelet-derived growth factor. Now, platelets are the little tiny bits. They're like bits of cells. They're not a whole cell. They're little bits that are used to help our blood clot. So when you get a cut, these platelets come in, and you can see here that they're starting to like pack together. They will pack together to like close up that opening, but they send out a ligand, okay, that stimulates cell division in the tissue around it, okay, that connective tissue, and allows us to heal wounds. Now, when we have those platelets plus some of the fibrinogen and the juicy stuff and all this that goes together, we wind up forming a scab. Okay, so that, I know it's weird, but that what is building that scab is sending signals to the tissue around it saying, we've got an opening here and you need to close it. So the cells start dividing and dividing and dividing and closing up that cell so that when that scab comes off, now you have skin underneath there again. Okay, it's really kind of cool how our body works. In some ways, it's like magic. Now, growth factors in cancer. Okay. Growth factors can create cancer. We have what is called a proto-oncogene. Proto means before, onco is cancer, and we know what a gene is. That's the part of the DNA. So it normally activates cell division. So proto-oncogenes say cells divide. Okay. It is a gene that gives us growth factors. They can become oncogenes if they're mutated. So if there's something wrong with this gene, if it becomes mutated, then it is no longer a proto-oncogene, it is an oncogene, which means it won't turn off. Okay, so it's turned on and it says divide, 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 divide. Well, that's not what our cells need to be doing. We have one that's called the RAS gene. Okay. Tumor suppressor genes work with the oncogene to turn off cell division. If we turn off this gene, it can't turn off the other. So that means when this is off, okay, it's not telling the proto-oncogene to stop. So we can have a mutation here or a mutation here. So this is a gene and this is a gene. If there's a mutation in this gene, then it's saying divide, 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 divide. And normally our tumor suppressor gene says stop dividing. But it can't keep up. If this is turned on, this and left on, this may not be able to keep up. Also, if this is on and this is off, that means it's not telling it to stop. Okay. Now we're going to talk about what causes these mutations in just a couple minutes. So a question was asked about healing. Wounds are healed. Why is it that they are never fully healed back to 100%? It's kind of like if you were to see, I don't know, let's say a car went through the wall here next to the school. They can come back and they're going to build it back. They're going to put, you know, the stucco, make it all built up how it was, but it's never quite the same because it's not the same age. It hasn't had exactly the same material, all of that. Plus, depending on the wound, if it has damaged any nerves, then we can have sensation difference, all kinds of things along those lines. So when we're talking about healing wounds, it's never quite the same because it's a patch job. We had a problem, we're patching it up, but patches are never quite the same as the original. It never is nice and beautiful like the original was. And it over time will fade and become more like the others. So you can get a scar and it may fade over time because the new cells that are being made are starting to replace even that scar tissue. That's why scars fade. So question is asked, why can't we do things like lizards' tails? Why can't we grow it back? We don't have the genes to do it. It's not in our DNA to be able to do that. So that's why they're studying it, is to see if they have a gene that can do it. Is there a way we could either use the mechanisms, if we could see what kind of chemicals are being produced or what is going on, or is there a way we could put the gene into another organism and have that organism be able to do that? So those are the kinds of things that they're studying. All right, go ahead and keep posting questions in the comments, guys, or in the chat. Now, our tumor suppressor gene, an example of that is P53, and this is a really common one to have as an example on the AP exam. So P53, we're going to talk about P53. Cancer is basically a failure to control cell division. So a cancer tumor, right? So a tumor is a lump. Well, if it hadn't grown and divided like crazy, it wouldn't have made that lump. It would have been normal. So when cell division is not checked and stopped, it grows like crazy. It is unrestrained and uncontrolled. Now, what control is lost? The checkpoints. Our checkpoints, we basically knock those gates down and it's like go constantly, no matter what's happening, whether the DNA is okay or not, all of that. Now, P53 gene plays a role in this first checkpoint, okay? It halts cell division if there's damage to the DNA. So if I stop P53 from working, that means that I'm not going to look for problems with the DNA. I'm just going to keep growing and growing and growing, including damaged DNA. And that's what we see in cancer. Okay, so P53 stimulates repair to fix the DNA, but if we break that, then it can't repair it. It will keep the cell from dividing. So if this chemical is there, it keeps the cell from dividing unless there's a way to clear it up. So if there's a problem, we put in P53. P53 says, stop, there's a problem over here, we can't keep going, and it will cause the cell to apoptosize, okay? Apoptosis, so it's gonna basically shrivel up and die. Now, all cancers, everyone that they've found has shut down P53 in some way. If it's an issue with lost control, does it involve phosphorylation? Well, they're trying to figure out exactly what it's doing still. That's still part of what they're studying with cancer, is exactly how all of these are working. They're finding definitely the P53 is not there, but they're not necessarily showing a difference in cyclin or CDK. So they're still working on that. Now, development of cancer. Okay, cancer develops only after a cell experiences about 6 mutation hits. So these are different types of mutation hits. And once it hits about 6 of these, it can't fight it anymore and cancer develops. So we can have unlimited growth. We turn on the promoter gene. We can ignore the checkpoint. Okay, we turn off P53. We escape apoptosis. So the suicide gene is turned off. We have immortality, where we turn on the maintenance genes. So it just keeps going and going and going and going instead of fixing and stopping, doing and stopping. It just is constant. We promote blood vessel growth. Now, cells have to get nutrients. And if a tumor has a lot of blood vessels growing into it, now it's getting like an unlimited buffet. It's all this nutrients coming in. And so it's going to grow like crazy. There's a gene that is triggered that turns that on. And then we can overcome anchorage and density dependence. So when we looked at our culture plate, it was flat, right? Just a flat layer of cells because it was density dependent. But in this case, it doesn't care anymore. And it's like, yo, just keep going, keep packing them in there. All right. So is the tumor taking nutrients from the body? Absolutely. The tumor is pulling nutrients out of the bloodstream, just like every other cell is. Can another cancer kill a different cancer? I don't know. I don't know if that's ever been looked at. It's another one I'm going to have to look into. So cancer v. cancer. I'll look into that one. So my derpy little penguin here says it's like a car with many systems failing. So sometimes if you have a car and there's one thing wrong with it, you can still kind of keep working, but you hit like a critical mass where you hit the point where you're like, that's just too much. Now it just doesn't work. The same kind of thing happens here. We can deal with one or two little problems, but once they start building up, then we can't deal with it. Then we have cancer. Now, what causes these hits? What causes these mutations? Mutations can be triggered by a lot of different things. One very common one is UV radiation. You guys know where UV radiation comes from? Yes. Yeah, you guys know. It comes from the sun. So UV radiation can actually, it is radiation, and it can damage the cell, and it damages the DNA, and it can damage the cell cycle. Something you may or may not know, when you get a sunburn, that's not a heat burn like if you've touched a stove. That's a radiation burn. Okay, that's why you don't feel it happening. And then later you're like, oh, that hurts. Now, I know some of you have beautiful dark skin and you're not pale like me, so you may not like have had that happen very often. But the damage done is radiation. We can have chemical exposure. When you're exposed to certain chemicals, it can trigger cancer. You may have seen advertisements for people who've been exposed to, say, asbestos, and that triggers cancer. The chemical is what basically winds up being like an artificial signal that causes problems in the cell. Radiation exposure, you've all seen the movies where somebody's exposed to radiation, they get sick and die. Well, that's because their cells are mutating and can't do what they need to do. Heat. You can actually, from excess heat exposure, you can have damage done to the cells and to the DNA, including mutation. Cigarette smoke, you guys all know, I guarantee you've heard it since you were very little, that cigarette smoke causes lung cancer. It has to do with the chemicals in there that are absorbed by the cells in the lining of the lung that causes mutation in the DNA. Pollution, this is again another type of chemical exposure. With high levels of pollution, they are finding a risk of increased cancer. And a lot of times that is, if it's air pollution, it's going to be lung cancer. If it's water pollution, you're looking at cancer at different parts of the body. And age. As we get older, our cells just don't do everything they're supposed to do. And sometimes they just kind of like can't. And so cancer can form. And then genetics. There are genes that make it more likely that you will develop cancer. Okay, there are genetics that can play a huge factor in this. This is why they say if you have a family history of any certain type of cancer, whether it's colon cancer, prostate cancer, or breast cancer, that if you have that in your family, you're more likely. Doesn't mean you will have it, but you're more likely. Now they have, we're looking at breast cancer here. We can see a tumor forms and it begins to invade the surrounding tissue. And then it loses its anchorage dependence and can actually float through our lymph and start forming tumors elsewhere. And it's the same tissue. So you can have a tumor form in your bone and it's still considered breast cancer. Now, breast cancer has a gene associated with it, one type of breast cancer, not all of them, that is associated with it. And if you have this gene, it means that you have over an 80% chance that this mutation will occur and you will get breast cancer. So over 80%. I don't know if you guys are aware, Angelina Jolie, her mother died of breast cancer. She got tested. She has this gene. So she actually elected to have a double mastectomy because it was either that or 20% chance she'd be fine, 80% chance she'd get breast cancer. So she had a double mastectomy and reconstruction. So now she's perfect because science put her there. Okay. So cancer is just a mutation in the cells that causes the cell to lose control. Yes, that's exactly what it is. So we have a mutation that causes that the checkpoints and signals for division to just no control over it. Good. Now a tumor is a mass of abnormal cells. So they're not the regular normal cells. It's a growth of them together. And there are different types. We have what is a benign tumor. Benign is fine. Okay. These are abnormal cells, but they remain at the original site as a lump, meaning they don't spread. P53 has come in and been like, we're done, stop. Okay, so it is like halted. Most are not a serious problem and can be removed by surgery. Sometimes these even happen and they don't even remove them because they're like, it's benign, it's not causing any problems, and it's actually more dangerous to remove it than to just leave it where it is. Okay, so benign tumors, benign is fine. Malignant, that's bad. Okay, malignant tumors are, they can leave the original site because they've lost attachment to nearby cells. They're carried in the blood and the lymph to other tissues. And if they start a tumor somewhere else, we call that metastasis. So if I were to get, let's say I got stomach cancer, and over time, it traveled through my lymph and through my blood, and it went to my collarbone, and I developed a tumor in my collarbone. That's called metastasis. So it's moved. It impairs functions of organs throughout the body. So you can actually have these float, and when they stick somewhere, they start growing there. So you can have cancer that travels throughout the body, and now all of a sudden you have tumors growing in the lungs, in the liver, in the pancreas, in the intestines, and it causes these things to start shutting down. Now, traditional treatments for cancer include radiation and chemotherapy. Now, radiation is a very targeted radiation. It's very pinpoint. So they usually will actually give the patients a little tiny tattoo for where they're directing the laser, because they'll have to do multiple treatments, and then they know they're getting exactly the right spot. Now, I said that radiation can cause cancer. Okay, so radiation kills cells, it can cause cancer. So the idea is to kill the cancer cells without causing anything else. Okay, they a lot of times won't do radiation on a patient that's really young, because 20 years down the line, they may develop cancer from the treatment. So they'll only do it if they really have to. They use chemotherapy, and chemo is chemicals, so chemotherapy. So it's a therapy using chemicals. And this can be done through IV. So sometimes they're IVs. Sometimes they are pills that you take. Sometimes they're shots. Okay. Medical radiation could cause a mutation later in life. It's possible, yes. So it's kind of balancing it of, is it the best treatment now versus what might happen later? So they balance that out. If they say that radiation is the best treatment right now, that means they've looked at those kind of odds and said, this is the best idea for giving you the longest, best life. Okay. And then do tumors have nerves? They don't, nerves are their own cells. So you can develop cancer in the nerve cells. So it's nerve cells that have mutated, but the cancers themselves don't have nerves. They can, as the tumor grows, if there's nerves nearby, they can push on those nerves and cause pain and things along those lines, but they themselves don't have nerves that are signaled to them. Okay. Now, what happens is we stop DNA replication, mitosis and cytokinesis, and stop blood vessel growth. Now, with chemotherapy, it's a race. We are killing cells. Unfortunately, when you put an IV in your body and it's pumping medication through there, when you take a pill, any kind of pill, it doesn't know where it's supposed to go. It goes into the bloodstream throughout the whole body. So we have to try to kill the cancer cells without killing the patient. One of the reasons for the rapid hair loss, one of the things you guys have seen is that people who have cancer lose their hair. It's from chemotherapy, and not all chemotherapies, but a lot of them, because they stop mitosis, they stop blood vessel growth, they stop DNA replication. That means it's also going to affect the cells that are making hair, and that hair will fall out. It's also not uncommon to have not just the hair on the head fall out, but like the eyebrows and eyelashes. All of that is affected. It can't do what it needs to do. It can't do its job. So there's nothing to hold it there anymore. A lot of times, once chemotherapy is stopped, once the person is healthy again, the hair comes back, not a problem. But sometimes the hair comes back different. For example, I have curly hair. If I were to have to undergo chemotherapy, when it grows back, it may grow back curly, but it might not. It might grow back straight. So there can be differences when it comes back. We're trying to kill that cancer, those cells that have that bad division going on, without killing the rest of the patient. So that's one of the reasons that people undergoing chemotherapy, they have suppressed immune systems. They have to be really careful about being around people who are sick. They have a harder time eating from side effects from the medication. The medication itself is causing all these problems to try to kill those cells, but we got to balance it with not killing the person. So it's a race between the medication killing the cancer and killing the patient. And, you know, basically we hit the point where we're like, okay, if we do any more of this medication, the medication is going to kill you instead of the cancer. So they're always balancing those things out. So when a doctor gives a course of treatment for cancer, it's because they've looked at all those things and said, based on what we've tested and had happen in the past and what we see going on with you, this is the best course of treatment. Now, what's really cool is that they are coming out with new drugs and new ways to treat it. Right now, we have what's called Gleevec. This is a targeted cancer drug. Okay, it's used to treat adult leukemia, so not the youth or child onset leukemia, and types of stomach cancer. It targets only cancer cells by adjusting, and Fiona, you mentioned the phosphorylation. So what it does here is it attaches where ATP would be, okay, for that phosphorylation. So now it can't do that job. It doesn't have the ATP to be able to phosphorylate. Okay, so Gleevec comes in here, and if it can't phosphorylate, that means it can't continue in the cell division process. Okay, so we stop that cell division. They are also working on other treatments that's still pretty experimental right now, but they actually will take a piece of that tumor and they will do a DNA sequence on that tumor, because it's going to be different than your DNA because it's mutated. And they are working on creating specific medication that targets only those cells. But if you think about that, that means that every single person that gets cancer is going to have a different medication that has to be made. So it's incredibly expensive. And again, it's still experimental. All right, we're almost done, I promise, guys. Here is a whitefish blastula. A blastula is after the egg has been fertilized. It now starts to divide. And in the very first stages, it's like a ball. We call that a blastula. So this is from a whitefish. Think of like cod, okay? And we're looking at that division inside the egg. You can see the different stages here. In interphase, it's just kind of chilling, kind of that, I don't know, fried egg kind of look to it. In prophase, it packs up our DNA. In metaphase, it lines it up in the middle. Now, I want you to remember these are three-dimensional, so this isn't really a line. It's actually a disc that's been turned sideways, so it looks like a line. In anaphase, this is early anaphase, it's starting to pull them apart. And if it pulls them apart more to be, this would be anaphase and early telophase, where it's starting to pinch in. And once it's done pinching in, we have cytokinesis and everything's divided. They're still packed up and haven't unspooled. That's how I know this is telophase and not just interphase. This is the same kind of thing, but it looks really cool. They've fluoresced, meaning they've added radioactive tags to it. So as they did the x-rays, they're able to, we can see different things happening. This here, that is the spindle fibers from our centrioles. Here we have it packed up here, here it's packed, it's been moved to the middle. You can kind of get an idea of how this is a disc when you look at it this way. And these are our spindle fiber threads attaching so it can pull them apart. Here's anaphase where it's pulled them apart. And then in telophase, you can see its cytokinesis is occurring and it's starting to divide into two separate cells. I think this is a really cool way to look at it. It makes it easier for me to understand what's actually happening. In plants, it happens a little differently. We have the same stages and everything, but plants have giant chromosomes. They're huge, really, really big chromosomes. So we can see them very easily. Here's metaphase, and see how, I mean that they line up in the middle, but they're kind of like, it might stick out a little bit. Here we have anaphase. It looks like a monster's mouth. Telophase. And this is the onion root tip. This is actually what we're going to look at for a lab. Now, I've been pushing off labs, hoping that we're going to be coming back in January and we could do them in person. Not sure if that's happening at this point. We'll have to see what the school board says. But in January, we're going to do some more labs. Hopefully you'll be here. If not, we're going to have to do them digitally with, I'm not sure how, simulations and videos and things like that. But one of the things you're going to do is look at onion root tips. So the tip of the root is where there's a lot of growth happening because the root is growing down into the ground. So as it's growing and moving down, we're going to see a lot of cell division happening. And this is kind of what it looks like. It looks almost like corn, like the kernels on a corn cob. Okay, that's because all these little boxes, these boxes are our cells. And you'll see some of them are smaller, some of them are gigantic. It depends on when division happened and when the cell wall was formed. We can see lots of cells that are in interphase. And you can see here, like this one has, this is a nucleolus where our ribosomes are made. And we have 3 of them in here. It has 3 nucleoli. Okay, so here's interphase. This would be prophase where we're packing everything up. Here's metaphase, it's lined up. This is anaphase. This is definitely anaphase. And it's actually the beginning. You can start to see a darker line happening here. That's telophase. You can see, where are my 2? Here we go. This is definitely in telophase because it's still packed up, but it's developing that wall there. So we know that that's telophase and cytokinesis. Okay. This is a lot of information, a lot. I know, and I'm really, really sorry. Do you guys have any questions? No, everybody's just fried. You're done with this. Okay, I really, really recommend taking a look at the handout 5 and doing that as soon as you can. Is cancer going to be a big topic on the test? It is going to be a topic on the test. It's a big part of understanding cell cycle, so it definitely is going to be in there. Is the test going to be morphed with the final? I haven't decided. I'm not used to working this way and having the final things are a little weird. I'm inclined to say they're going to be 2 separate tests given during that hour. Okay. There will be FRQs on signaling and cell division. The final is 2 hours per period. Well, 1 hour and 50 minutes. So like 8 to 9:50, 10 to 11:50. 2 hours for each final. Okay, I will make sure that these get posted. I did post the notes for the 4-1 in the YouTube channel. If you can't get to that, let me know. I'll see if I can download it or something for you. But you should be able to get to it through the YouTube channel. If you need that link, it is in the class announcements. Look down at the very bottom of the classwork page. FRQ rewrites, I'm hoping to have those posted tomorrow. The final will have multiple choice and FRQ as well. You can expect the FRQ portion on the final to be primarily things like math and graphing. All right, with that, I will see you guys tomorrow. Don't forget that you have a... Bye, guys.